Right, cell-mediated immunity. All right, what does that mean? IL-2 causes T cell proliferation. So it helps an activated T cell make more of itself. Right? TNF does a lot of things. Increases vascular permeability, increases expression of adhesion molecules. All of that is important for inflammation, right? And gamma interferon activates macrophages. And an activated macrophage is a pissed off macrophage. So anything that a macrophage can do, an activated macrophage does way better. So an activated macrophage, as you'll find out when we get to the micro part, a lot of microbes think they can, and they do, replicate very nicely inside macrophages. So what does the macrophage do to try and kill them? Gets activated by gamma interferon secreted by Th1 cells. So inflammation, T cell proliferation, activation of macrophages. Important functions carried out by the cytokines
of Th1.
Th2 cells secrete things like IL-4. Wait, IL-4's up there. I know. It's just the way it is. IL-4 causes a naive T cell to become a Th2 cell, which then secretes IL-4. Go figure. But 4, 5, 6, 13, 10. A lot of these are involved in B cell activation and proliferation. Well, let's see, B cell activation, differentiation, meaning that the B cell has to become activated, has to differentiate into a plasma cell. Well, first it proliferates, then it differentiates. Okay? And then the Th17 cell secretes, not surprisingly, what? 17. It secretes IL-17. That's why it's a Th17 cell. IL-17.
Is that IL-23 too?
Yeah, IL-23 also. Sort of indirectly, and that won't go through these dashes because it is 7 o'clock, but indirectly leads to an increase in neutrophils in the blood and activation of neutrophils, so they're more unstable, they're just pissed neutrophils, and activation of neutrophils and chemotaxis, or the attraction of neutrophils. Now, indirectly, that's what that means. Indirectly, IL-17 does not directly cause neutrophils to be produced in the bone marrow. There are other cells that are stimulated that are going to release other cytokines that will blah, 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 blah. It's very complicated. But just remember, it's indirectly leads to this. So
it increases neutrophils, activates the neutrophils, and chemotaxis the neutrophils.
yeah. So if you have a response that's really reliant on neutrophils, you need a lot of neutrophils to get rid of something, the Th17 cells are the way to go. If you need a lot of antibody being produced to fight the infection, Th2 cells are the way to go. They're also good for parasitic infections. worms. And then if you have an intracellular pathogen, an intracellular pathogen, Th1 cells are the way to go. So intracellular, extracellular, and actually these two are extracellular. Intracellular pathogen, extracellular pathogens.
Wouldn't know like Th2 is part of anti-inflammatory.
Th2s, yeah, well, yeah, they can downregulate activity of Th1s, but that's going into more detail than we need to do tonight.
Dr. Buxton, did you say for Th1 differentiation, you said that you need IL-12, and what else do you need? You said
IL-12. For Th1s, IL-12.
just IL-12. How about Th2, just IL?
Th2 is just IL-4.
Okay,
Then for 17s, you need TGF-beta and IL-6
all together.
together.
Okay.
Yeah.
And then what was VP again? I'm sorry.
BP.
Right there. Inflammation.
Oh, vascular permeability.
Okay, and AM
And
is.
in adhesion molecule
Oh, okay.
expression. Yeah.
Okay, are we done?
Th1s do what? Intracellular.